Hello, I'm Kristi Gladem.
And I'm Kelly Hanrahan. We are from the Colorado Department of Agriculture, Palisade Insectary.
In this presentation, we will be discussing updates on Rearing Ceratapion basicorne, a rosette feeding weevil for use against yellow star thistle.
Yellow star thistle is a winter annual native to the Mediterranean basin. It was introduced to the U.S. in the 1800s. Since its introduction, it has invaded over 41 states, but is most common in Idaho, Washington, Oregon, Utah, New Jersey, and especially California. This highly invasive, economically and ecologically damaging weed has been the target of biological control efforts since the 1960s. 7 species have been introduced to attempt control of yellow star thistle populations. All of these species are seed head feeders that reduce the reproductive capability of the plant, but so far have not provided a high level of control. New biological control options are needed.
Ceratapion basicorne, a root-feeding weevil, was first proposed as a biological control for yellow star thistle in the 1980s by Clement. However, forced choice laboratory testing showed some feeding damage on safflower, so it was not pursued further at that time. In the early 2000s, Dr. Lincoln Smith with USDA ARS revived Ceratapion basicorne as a possible control and established a laboratory colony from weevils collected in Turkey. This Turkish colony resulted in the majority of published work on this weevil. In 2009, U.S. release was denied and the colony was terminated. However, in 2019, Ceratapion basicorne was approved for release in the U.S. and a new colony using Greek specimens was established in Albany, California.
Ceratapion basicorne, also known as SIBA, is an incredibly small weevil measuring approximately 2 millimeters in length. They are univoltine, meaning they have only one generation per year. In early spring, adults emerge from their winter diapause and will begin laying eggs within the leaf tissue of young yellow star thistle rosettes. The larvae mine through the main stem of the leaves into the taproot where they will pupate into adults. Development of the weevils is highly correlated with the yellow star thistle plant growth. Pupation occurs as the plants bolt, and the new adults emerge as the plants flower and senesce. New adults require a period of warm temperatures followed by a winter diapause to become reproductively active and begin the cycle again the following year.
In 2022, the Palisade Insectary received 10 SIBA females from the Greek colony established at USDA ARS in Albany, California. These females were isolated directly on live plants to oviposition in the leaf tissue. The ladies were moved 3 times per week, and we recorded feeding holes and numbers of eggs laid. After an average of 67 days, the new adults emerged. Only 5 of the 10 females reproduced successfully, and 49 out of 101 plants exposed resulted in new adults. Of the resulting 84 weevils, 39 were from a single mother, our champion weevil. We additionally experimented with some larval transfers, and in total, we reared 95 adults as the first generation of our colony.
In Colorado, yellow star thistle is a List A invasive species, with fewer than 11 known populations in our state. To grow these plants for SIBA development and feeding, we carefully track each seed. Seeds are germinated on filter paper before planting in soil. The plants must be grown for 10 to 12 weeks before they can be infested with ceratapion. At the Palisade Insectary, we are able to utilize greenhouse space and continuously produce yellow star thistle throughout the year. For 2023, we are aiming to produce over 1,000 yellow star thistle plants. Our 2023 rearing process is a bit different. We begin by removing a portion of our G1 population from winter conditions. If they have successfully made it through estivation and diapause, we expect them to begin ovipositioning approximately 3 weeks after being brought out to ambient lab conditions. We hold each weevil individually with a cut yellow star thistle leaf. The leaves are changed and checked twice per week. Leaves exposed to the weevils are carefully looked at under a stereoscope. The photo on the right shows numerous feeding holes within the blue circles. Inside the red heart is a shadow cast by a SIBA egg laid deep within the plant tissue. For 2023, we are aiming to have 10 females actively ovipositioning at a time until all of the G1 have reproduced.
When eggs have been identified, we delicately dissect the leaf tissue to expose each egg. We then carefully remove the egg from the leaf using a fine paintbrush. We transfer the eggs onto wet filter paper in a petri dish for development. This method allows us to track each stage of the egg development. In the larger photo on the right, 4 stages of egg development are seen. Eggs are bright yellow and a solid color from days 0 to 3. From days 3 to 6, the casing of the egg darkens and embryogenesis begins. The head capsule of the larva develops during days 6 to 8, and then the larva is ready to eclose 8 to 13 days after being laid. In this video, a larva is actively eclosing and freeing itself from the egg casing. After freeing itself from the egg casing, the larva actively searches for food. These larvae, though just a half a millimeter in length, will travel quickly and far in their search for food and must be checked on several times per day. Once they are fully eclosed, they are transferred onto a live yellow starthistle plant.
For this rearing protocol, we are only utilizing live larval transfers. This allows us to ensure viable eggs are being laid, larvae have successfully eclosed, and only 2 individuals are allowed to develop in each plant. Once a larva successfully ecloses, a healthy yellow starthistle plant is selected for the transfer. A single leaf is isolated, and a scalpel is used to create a V-shaped notch in the petiole, a couple inches from the plant base. The neonate larva is carefully picked up with a fine paintbrush and placed in the exposed inner tissue of the leaf. Leaves are marked and later checked for larval tunneling, which shows a successful transfer. These plants are labeled and kept at ambient lab conditions. After 6 weeks of development time, each plant is sleeved to prevent adult escape. Each plant will be checked weekly for adult emergence.
Ceratapion basicorne is a very new biological control agent to North America, and many mysteries are still unanswered about this cryptic weevil's life cycle. We have several goals and questions for future study. First, we would like to improve the mass rearing protocol, increase efficiency, collect better data, and produce a large enough G2 population to conduct field release studies and laboratory studies. The main focus of our laboratory studies will be to find minimum estivation and diapause requirements through temperature and photoperiod, as well as response to food deprivation. Long-term goals include using the Greek colony to replicate studies done with the Turkish colony, which represents the majority of literature on this insect. We would also like to acquire SIBA from several geographic regions in the Mediterranean to study the genomics and population diversity of this species.
This project has been made possible through the many decades of work accomplished through several agencies.
We want to extend our gratitude most especially to the following individuals. Thank you, Dr. Lincoln Smith, Dr. Brian Rector, and Irene Wabawa at USDA ARS.
Additionally, we would like to thank Drs. Dan Bean and Tom Dudley for their continuous support of this project.